Hey Lauren, how you doing?
Hi, how's it going?
Sorry my camera's broken, but I'm looking forward to your talk.
Thanks.
Okay, Lorna, you're there.
Yep.
Cool. Okay, I guess we'll get started. So hello, everyone. We have some people still coming in, and we'll let them in as the talk goes. But okay, Lorna, welcome everybody to our second NextGen seminar, epilepsy seminar. We're very glad that you're here. Today we have Lauren Lau with us, and Lauren is a postdoc with Kevin Staley at Mass General in Harvard. And Lauren has been working in the epilepsy field for some time. She got a BA in biology at Boston University, and then moved to Tufts, staying in Boston, and worked with Chris Dula. Worked in epilepsy, worked on the freeze lesion model and thrombospondin signaling and neocortical circuits. And then she moved over to Kevin Staley's lab at Mass General for her postdoc appointment, where she's been working on organotypic slice culture models and network activity that leads to seizures. And so Lauren, we're very glad that you're here. I've opened up the chat this time, so if you have questions for Lauren, you can write the question in the chat, and I can read it back to her later, or at the end of the talk, we'll open it up and you can unmute yourself and ask a question if you want to. Okay, so with that, Lauren, the stage is yours.
Well, thank you.
We'll open it up.
Okay. Sorry, there's feedback from in. All right. Well, thank you to Bobby for organizing this seminar series and for inviting me to speak today. And I'm excited to be sharing this work with you, which is really focused on looking at seizure onset at cellular resolution. And to dive in, I kind of want to start by posing a fundamental question in seizure research, which is how does the epileptic brain transition into seizure activity? And we can think about this in the context of acquired epilepsies, where there is an initial precipitating injury, a period of epileptogenesis, and then a stage of chronic epilepsy characterized by recurrent spontaneous seizures. And I'm sort of showing here in the red arrows, we have these different discrete seizure events. So even in the stage of chronic epilepsy, there's transitions between physiological activity and seizure activity. And I like to think about it in this way, that for many patients with epilepsy and in our animal models, the majority of the time is really going to be spent in the interictal period, generating different patterns of physiological activity. And sometimes from these states, the brain may move into a proictal state. And this is a place from which it becomes more probable to trigger a seizure, but is not definitely set the course. And you may transition between these two different brain states before finally the occurrence of ictogenesis, in which there's sort of been determined enough changes that have built up that you're now going to trigger a seizure. And so really, I'm going to be focusing this talk on this concept of ictogenesis. So what defines this transition to the seizure state? And we can think about this on the cellular level to say, what is the cellular substrates of ictogenesis? Is there a particular population or subset of neurons which are really critical for driving this transition? And one candidate is the class of interneurons. So we have several studies which have shown that interneuron activity precedes seizure onset. This has been shown in chemically induced seizures in vitro models and in some in vivo studies in both animal models and in epilepsy patients. For example, this was a study in epilepsy patients, and this was from unit recordings using spike sorting to look at the activity of putative interneurons versus excitatory cells. And what you see is the 0 time point here is seizure onset, and we see this pink line is the interneurons, and their firing is increasing prior to onset and peaking right at the end of the at the beginning of the seizure onset, whereas the excitatory cells activate a little bit later. Another hypothesis for kind of a key driver of transition to seizure has been the idea of hub cells. So hub cells are highly connected GABAergic interneurons that have been identified in the developing hippocampus. So in this figure, the red is showing the axonal structures of this hub cell. So they're much more highly connected than a typical interneuron. And early in development, it's known that they have high connectivity. So on the right-hand panel, when this single hub neuron is stimulated, you get activity in a large number of neurons, so more than you would expect in a typical interneuron. So it's been suggested that these hub cells may play a role in driving seizure initiation because we know that they persist into adulthood, at least structurally. So it's unclear if they have a role in the adult hippocampus normally, but again, because these highly connected cells are still in the hippocampus, they may get reactivated in a pathological way during seizure. But another type of connectivity that we can think about in the context of recurrent spontaneous seizure is small world networks. So in this type of network, neurons will primarily have local connections with just a few long-range connections. And it's been shown that this type of connectivity map can engender seizure activity. So this is examples from a couple different computational models. And by varying the degree of long-range versus short-range connections, you get different types of activity. So for example, if all of your connections are local, then you get low-level activity that does not propagate through the network. If on the other end you have random connectivity, or to say, primarily long-range connections, you get spiking, but not necessarily seizure activity. And it's really in this range of small world connectivity that you get seizures being generated. And again, in this type of network, there's no particularly high connected neurons that are sitting in a privileged position, but rather just this underlying map is able to induce seizure activity. And some modeling from our own lab has expanded upon this type of small world connectivity to show that you can have a network that spontaneously generates both spikes and seizures dependent on the amount of available glutamate. And so having a partially refractory small network will allow for recurrent spontaneous seizure. And so we can see that there's a couple different candidates for how these seizures may be beginning, but really we haven't been able to address it experimentally because we really need cellular resolution techniques. And this has been challenging to study for a number of reasons. The first is that by definition, seizures are spontaneous. And so particularly because we are focusing on the initiation phase of seizures, we don't want to be evoking them with chemoconvulsants or by altering the extracellular ions. And so really we want to only look at spontaneous seizures. And so to capture those, you need very long recordings, which can be challenging for technical and practical reasons. Additionally, in vivo, it can be difficult, if not impossible, to separate the differences of seizure onset from seizure propagation. So in a situation where you have a field of view or in electrodes in one area of the brain, when you first detect the earliest pathological activity, that may be the seizure onset, or the seizure may have began in a different brain region, and now you're just again seeing the propagation into your field of view. And finally, a lot of our techniques that are available to us have rather poor spatial resolution. So you are either looking at population means of activity, or you're going to be kind of undersampling and looking at just a handful of neurons and trying to build the whole picture of what the network is doing. So to overcome some of these challenges, I have been performing chronic imaging in hippocampal organotypic slice cultures. This is a model that our lab has been using for a number of years. And in this way, you can create hippocampal slices and maintain them in culture for days and weeks at a time. And in this case, the actual slice preparation is the injury. And after that, we don't do any further perturbations to elicit spikes and seizures. So this raster plot is showing the electrical activity from a multi-electrode array. And you can see that in the kind of early days in vitro, the slice culture generates a lot of spikes, and later will start to generate seizure-like events. And so what I've done is to switch to an all-optical method of looking at activity in these organotypic slice cultures. We have a custom developed imaging system, coined our IncuScope, where we can leave these slice cultures in the incubator and periodically position them for imaging, and leave them undisturbed in a way that allows us to track the activity of individual neurons over time. In this work, I've looked at calcium activity with a GCAMP7 fluorescent protein, and this is expressed under a synapsin promoter. So we have pan-neuronal GCAMP expression, and this is to kind of give you a sense of what that expression looks like. This is a standard deviation projection to kind of highlight the active GCAMP positive cells. In these preparations, I'm also labeling the interneurons. So this is an example of TD tomato labeled interneurons under a DLX-Cree driver. And I'm going to play this through so you can see what the interneurons look like over time, and to really highlight the stability of these slices in culture. So I'll let that loop through one more time, and you can see that the overall anatomy of the slice is very stable. There's a little bit of cell loss over time, but not a lot. So this is a really nice model for us to start to look at what is happening with the seizure activity on a cellular level over time. To first kind of give you a snapshot of what the seizure activity looks like in these slices. We performed low-powered optical field potentials. So this is a low level of illumination to look at just the mean fluorescence of the slice over time. So you can see, for example, what our seizure-like events look like. So they're tens of seconds long, high amplitude, polyphasic, generally with a postictal depression. And we can also look at this over time in this raster plot. In this case, each row is 1 day, and it's showing an hour's worth of one of these optical field potentials, with the color bar showing you the amplitude of the calcium signal. So each one of these yellow periods is one of our seizures. And you can see just by eye that they begin to emerge after a few days in vitro, and they increase in frequency. So this is the quantification of that across many slices. Again, over the course of about the first 2 weeks in vitro, we see an increase in the frequency and duration of seizure. So now we really want to dive into our ability to look at the activity of these cells at cellular resolution. So I'll play this through so you can get a sense of what the calcium activity looks like in these slices during a seizure. And from these types of movies, I performed an automated ROI selection. So this relies on the standard deviation projection to, again, really pull out the active GCAM positive cells. And then we can use some algorithms to automatically detect all of the cells in this slice. And one challenge from this type of imaging is because we have wide field single photon imaging, we do get a fair amount of neuropil contamination. And what we can do to correct for this is to really calculate a local surround neuropil and perform a correction so that we can really start to isolate the calcium traces of individual neurons. And so this is just kind of a closer look at what we can kind of generate from our average calcium signal across all of these neurons. And this is with a paired LFP. And again, we're really focusing on this initial onset period of the seizure. And so I've defined this as the earliest detectable rise in the mean calcium. And then it is kind of focused right before the onset of frank seizure, which is characterized by high amplitude hypersynchronous calcium. So you can see just an expansion on that ictal onset period within this seizure, and also just kind of zooming in on the other types of tonic-clonic-like bursting later in this seizure. And so as I started to kind of collect a lot of these different seizures, the first thing that became very obvious was that there was different types of seizure onset in the hippocampal slice cultures. So the first category that we defined was low amplitude fast activity. So this is the same seizure as the previous slide, and this type of onset is going to be similar to, for example, a low voltage fast onset, and it's really characterized by several seconds of low amplitude activity building up to the transition to frank seizure. The second type of onset that I observed was sentinel spike onset, and this is defined by a very brief onset period, less than 2 seconds, and you really have maybe a single spike or a few spikes before you have the transition to frank seizure. And the final type of onset that I observed was spike burst plus low amplitude fast. So in this type of onset, it's really initiated with a spike, a high amplitude calcium spike, which in the LFP is this sort of barrage of electrical activity, and then it's preceded by several seconds of low amplitude fast activity before the transition to the full seizure. And so I'll just give you a chance to see an example of each one of these types of onset. This is an example of low amplitude fast onset. Again, really characterized by this buildup of activity before the kind of full activation of our entire network with high amplitude hypersynchronous activity. This is an example of sentinel spike onset. And really that's, you know, the most obvious feature of that is just how quickly you go from the brief onset into the full seizure. And I'll just give you the chance to kind of see the whole seizure. And then the last example is of our spike burst plus low amplitude fast. So that was the high amplitude burst of calcium activity, several seconds of low amplitude, and then transition to full seizure. And what was very interesting was that there was a really characteristic evolution in the types of onsets that were being generated over time. For example, in the first few days that seizures were being generated, they were all of the low amplitude fast type. And later on, the slices would also generate sentinel spike or spike bursts plus low amplitude type seizures. And interestingly, even within individual slice cultures, they didn't kind of switch from one type to the next in an absolute manner. So even within the same day, in the same exact slice culture, it might be generating a sentinel spike seizure and then next low amplitude fast and kind of back and forth. So there were sort of different time periods where certain modes of onset were more likely, but again, it didn't switch in an absolute way, kind of suggesting this dynamic changes in how the seizure initiation was occurring. And the sentinel spike and spike burst seizures, at least in this model, seem to correlate with the more kind of mature seizure phenotypes, the more severe occurrence of seizures. So this is the quantification of the mean interictal interval. So as I showed before, frequency of seizures increases in this model. So in the later days, we have a shorter interictal interval, and this correlates with having a higher proportion of sentinel spike or spike burst onset seizures. And this is also quantified on a slice by slice basis. So you can see that on the days when only low amplitude fast seizures versus mixed onset or just spike burst and sentinel spike, there was a significant difference in the interictal interval. So in addition to seeing the kind of development of different types of seizure onset, I also saw a refinement of the seizure onset zone over time. So in this figure, the first panel is just showing you one of our DLX TD tomato labeled slices because it's a little easier to see the anatomy of the slice with the labeling. So you have, dentate up here, highless, CA3, CA1. And interestingly, when these slices were first beginning to seize, for example, you can look at this one from DIV5, there was generally not a well-defined seizure onset zone. So looking at the anatomy of what are the earliest versus later activators, it seemed to kind of have a more general activation across the slice at seizure onset. Whereas at later days, for example, in this slice at DIV10, there was a really clear regional specificity of what were the earliest activators versus later. And this was true across all of our slices. So this is looking at the proportion of slices with a regionally undefined seizure onset zone. So you can see in the beginning days of seizing, all of the slices kind of had an undefined onset zone. And by the second week in vitro, there was kind of a refinement to a clear-cut seizure onset zone within the hippocampus. And once that seizure onset zone was sort of solidified, at least in this model, it seemed that all of the seizures began with activity in the CA1, often also with activity in the dentate and highless. And also in general, once a onset zone was developed, it was very consistent. So for 60% of our slices, they had only one seizure onset zone type, and this was consistent even across the different types of onset. So low amplitude fast sentinel spike, they always would begin within the same slice from the same regions. And you see that few of our slices had 2 or even 3 different types of seizure onset zones. So kind of the first conclusion from this is that using these long-term recordings, it became obvious that there's an evolution in both onset type and the regional onset zones. And I think that's really just to highlight that epileptogenesis is not necessarily over after the first seizure, and that these networks are continually changing even during the phase of chronic epilepsy. So for the next section of this talk, I really want to now focus on using our cellular resolution imaging to really start to dissect what goes into the different activity changes during the phases of onset and ictal activity. The top plot here is showing the mean delta FRF calcium. And again, I'm really zeroing in on this onset phase. So this is what you could kind of glean from population level changes. But in this case, from this particular slice, I had over 1,000 individual neurons that I can look at their activity and see, really how is this leading to the population level changes. So again, each row here is a single cell over about 45 seconds. You can see kind of the heterogeneity in their calcium activity over time. And one way that we, I think about how population levels change is basically to say, is a cell active or inactive? So in this case, I've set a threshold on a cell-by-cell basis to say, kind of convert that activity into a binary above or below threshold. And in this way, we can plot the fraction of neurons above threshold. So as expected, we see that during the onset period, there's an increase in the number of active cells, and we're looking at this as our measure of synchrony. So we have an increased synchrony during the onset phase, and especially at this transition to frank seizure. And you may imagine that could kind of explain the whole story, that population level changes are really an average of who's active and who's inactive. But what we were able to see is that in addition to the changes in the number of active cells, that actually the intracellular amplitude of neurons also changes over time. So this bottom plot is looking at the mean amplitude now of just the cells that are already over threshold. And so that's calculated on a time-by-time point of which are the active cells, but you can see that on average during the onset period, there is an increase in the intracellular amplitude. And so by having the information for these individual neurons, we can really quantify the contributions of these two sources. So how much of the mean population rise is due to synchronous activation, how much of it is due to these changes in intracellular amplitude. And so here I've quantified that across all of our recorded, in this case, low amplitude fast seizures. And you can see that during the onset phase, you get about 28% of this signal is due to synchrony, and actually a really large portion of the population level changes is really due to the rise in intracellular amplitude of the calcium signal. And the same sort of quantification can be done in the ictal phase. So again, to say this rise in mean calcium, how much of it is due to synchrony, and during the seizure itself, we can see that the majority of that signal is really due to synchrony and coactivation of many of the neurons at the same time, along with some of that coming from the amplitude. And sort of the final part in considering this question of how this signal is increasing is to separate synchrony versus recruitment. So jumping back here, you may imagine that as the fraction of active cells increases, and particularly with this large jump at the transition from onset to ictal, that you may imagine that this is largely due to a brand new population of cells being recruited at this next phase of activity. But interestingly, I found that was not really the main driving factor in the increases. So this is a plot from the same example seizure. This is showing kind of total newly recruited cells over the total number of cells. And you can see kind of recruitment is ongoing during this onset period. And there's a little bit of new recruitment at the transition to seizure. But it's particularly obvious when you look at the blue line is now showing you the cumulative on recruitment. So you can see by the end of the onset period, in this particular example, over 80% of the neurons have already been recruited, which is to say they've already been activated at least once during this onset period. And it's really only, in this case, the last 10% or so of neurons that are newly recruited at the end of the onset period. And I've quantified this across many seizures of the 3 different seizure onset types, and that seems to be universally true that we have a large amount of the recruitment occurring during the onset period, and particularly in low amplitude fast and spike burst. Almost the entire network is activated in the onset period. And so another way to look at that data, this plot is again showing the fraction of neurons above threshold, and the color bar is now letting you see what proportion of those active cells are newly activated, what proportion is due to recruitment. And sort of by definition, at the beginning of the onset phase, it's mostly new recruitment of cells, but later on you can see that the total number of cells active is really mostly cells being reactivated, and that there's not a lot of new recruitment at the end of the onset phase and into the seizure itself. And I've also quantified that across our 3 different types of seizure onset, and it seems to be fairly consistent. So kind of having this ability to really have cellular resolution detail, we see that the seizure onset is characterized by low-level changes that happen in a large population of the cells, and that really recruitment is largely finished by the end of the onset phase in these slices. So in the next section, I want to kind of break apart what is the contribution of principal cells versus interneurons. And here I do that by using our DLX pre-labeled interneurons. Again, we have GCAMP7 under our synapsin promoter, and for the purposes of identifying interneuron versus principal cell here, all of my GCAMP positive, TD tomato positive cells are being considered interneurons, and GCAMP positive, TD tomato negative are principal cells. And so to first break this down, just to see what the activity of these 2 populations of cells are kind of far away from the onset of a seizure. In these slices, there's relatively low activity in between seizures, but overall there was no difference in principal cells versus interneurons. So then I can look at what is happening during the onset phase. And so this is just quantifying the percentage of either principal cells or interneurons that are active during onset. And as I showed in the previous slide, most neurons, regardless of cell type, are activated in this phase, but there is a slight increase in the likelihood that the interneurons are activated during onset versus principal cells. And finally, I also plotted the onset times of principal cells versus interneurons in these cumulative distributions. And for each one of our seizure onset types, we see a slight leftward shift, which means that interneurons are activating earlier than principal cells, but this is on the order of milliseconds, and it's really, I guess, a pretty subtle difference when you look at these 2 populations as a whole. But it is consistent with interneurons being more active during ictal onset and activating slightly earlier. This was, again, a pretty modest difference when you look at these populations as a whole, but it kind of led us to question if there was a subset of interneurons perhaps that were very critical and were the early activators. By looking at the total population as a single average, you may kind of be missing some of the effect. So in this final section, I really want to take advantage of the fact that we were able to do chronic recordings and record multiple seizures and track the activity of individual neurons over time. So this is showing the onset rank of neurons across 6 different seizures. In this case, seizure 1 is our reference seizure. So the onset is, they're sorted by the onset here. So we have the earliest activators down to the latest activators. And the black bars are for neurons that were not detected by the automated ROI selection in that given seizure. And then for the subsequent seizures, the individual neurons are ordered in the same way. And you can see what is their rank in seizure number 2 and seizures 3 and so on. And I think even just by looking at this by eye, you can see that there's a lot of variability in the sequences of neuronal activation seizure to seizure. And I've quantified this using a Kendall rank correlation. So for example, seizure 1 compared to itself will be a perfect correlation, but you have seizure 1 compared to seizure 2 compared to seizure 3. And when I did this type of quantification across all of our groups of seizures and all of the types of onset, it was very consistent that we saw a very low correlation. So it is a positive correlation, but again, quite low, suggesting that there is not a repeatable sequence of neuronal activation that's being triggered seizure to seizure. And that's not to say that this is, there's no organization into how the seizures are beginning. So this is showing for each one of these neurons above the mean onset rank and where they are in the slice. So you can see that there are regions of early activators and late activators, but it's really when you start to break it down cell by cell that it's very stochastic. And so in looking at this type of quantification, it's comparing the entirety of the sequence seizure to seizure. So you may also imagine that maybe after the initial phase of onset, what happens after can occur and cells join in different ways, seizure to seizure, but maybe we've missed the critical drivers of ictogenesis at the beginning of the seizure. So kind of a final way to quantify the onset ranks across seizures is with this cumulative distribution method. So here, the multicolored lines are the distributions of the onset rank seizure to seizure. And so when a single seizure is being looked at, obviously they're always going to start at 0. So this is a normalized onset rank, and every seizure has to have a neuron that was the first activator in that seizure. But then what I did was to say, okay, this individual neuron, what was your average onset rank over the 6 seizures? And that's what is being plotted in the black line. And here you can see that this cumulative distribution of the average starts at 0.1, which is to say that no individual neuron fell into the top 10% of active cells over multiple seizures. And in other words, every seizure began with a slightly different population of neurons as the earliest activators. And you can compare this to the blue line here is the distribution you would get if you had 6 totally random time series that were shuffled and averaged together. And if you had a randomly generated series, what would you sort of by chance expect the minimum to be? And you can see that it's quite close to the experimental. And I did this same type of analysis across all of our slices, and you can see that the multi-seizure mean compared to a paired number of randomly shuffled series, the minimum from that, and you can see that they're quite similar. So it's really is suggesting that a different neuron or different sets of neurons are the earliest activators seizure to seizure. And the final kind of part of this that I considered was that we may have missed a critical driver of initiation just based on where we set our window. So here, again, seizure onset is being determined by a detectable difference in the population mean. But you can imagine that if there was a small number of cells that were very active prior to that, we would have missed it if we were only looking kind of a couple seconds later. So I also looked at the activity of neurons in kind of a 5-second preictal period to ask the question, is there a subset of neurons that are consistently highly active right before the seizure onset phase? And so this is showing the average normalized activity in that time window. And what you can see in these histograms is that across all of our different onset types, there's a high number of neurons that are consistently silent in the seconds immediately prior to seizure onset, but that you can see it on this end, and it's actually cut off at 0.8 because there were no neurons that were consistently highly active prior to seizure onset. And I think really looking at all of this data together really suggests that ictogenesis is not being initiated by hub cells or some other kind of small population of neurons. Yeah, and so with that, I really want to kind of bring it back to the question I posed at the beginning, which is, how are these transitions occurring from interictal to seizure? And kind of the main conclusions from this work is that seizure onset is really characterized by low-level changes across a large population of cells. We do have interneuron activity preceding that of principal cells to a modest degree, but that really when you start to break things down on the cell-by-cell level, you really see this stochastic variable recruitment seizure to seizure, which really, I think, highlights that each one of these seizures is being initiated along non-unique pathways. And so I certainly haven't answered the question I've posed, but I think it can begin to frame how we think about this, and that it's not going to be as simple as a couple of bad actors, and if you could find these hub cells and remove them or silence them, you would be able to stop the seizures in this network. But rather, there's probably more general modulations that are pushing perhaps the entire network closer to a firing threshold, and that once it's in this state, it doesn't quite matter which are the first cells to cross threshold, but that you can get these reentrant seizures over non-unique pathways. And yeah, so I will end there. I would love to take some questions, and I'd also like to thank my lab, particularly Kevin Staley and Kyle Willis, for their help in having this project go.
Awesome. Thanks, Lauren. That was a great talk. Can you hear me?
Yes, I hear you.
Okay, cool. So that was a great talk. Really interesting. We have some time for questions. If there are any, you can unmute yourself if you have a question, or you can write it in the chat if you want to, and I can just read it to her. Okay, we have a question from Laura Ewell. And she says, do you ever see seizures aborting? In other words, dynamics that mimic the pre-ictal to ictal transition, but don't develop into the full-blown seizure?
Yeah, so we do see that, and it's something I'm interested in kind of following up. So we have, I guess, two classes of activity that I would say is similar. So we have ones that are more traditional interictal spikes, and so I think we have kind of some ideas to compare what happens during a spike versus a seizure. But even in between, we have some activity that's I kind of just consider epileptiform. It's not quite long enough to be a seizure. And those tend to be very similar to how some of these seizure onsets look. And so, yeah, that's something I see, and I think could be a great place to start to say, why did that activity die out and in the seizure it did not?
Cool. And Laura's giving you the thumbs up, so that's good.
Okay.
Okay, second question. Elliot Smith says, sorry, I may have missed this, but are the onset rank results consistent across different onset types?
So I've mostly broken it down to keeping the onset types, comparing them to themselves, and those are already variable seizure to seizure. I haven't done it kind of in a comprehensive way to compare within the same slice different onset types, but just by spending some time with the data, I think it would be just as variable, if not more, to say what is the onset ranks of this group of low amplitude fast and then sentinel spike in the same slice. So I haven't done that, but my sense is it would be just as variable, if not more.
Cool. So we have another question that just came in. Jan Frankowski, so a graduate student in my lab. Would you expect the activity of VIP interneurons to precede those of the other interneurons?
So I don't know if I would have kind of any candidate class that I would think precedes it. And so, we, did the first kind of just looking at all the interneurons together because we kind of were thinking that we would see some subclass and we could then use the anatomy and morphology to kind of make some guesses about subclasses and go from there to kind of really start to dissect which are the critical ones. But what really came out of it is that, the interneurons are active a little bit earlier, but it was on kind of such a small scale. And when we had, in these onset ranks and where I'm looking at them seizure to seizure, here I haven't separated it out by interneurons versus principal cell, but I have done that, and there's not a sense that there was a subset of interneurons that were much earlier than others or that were much earlier than the principal cells. So I'd say it's not impossible that a subclass of interneurons may be a little bit earlier, but just kind of looking at the whole picture, it doesn't seem like that'll be a strong candidate for explaining the entire, that this group activating first is what really drives the onset.
Okay. So we've got a bunch of questions coming in now. So next one's up, Karen Wilcox. So Karen says, great talk, gorgeous data. I might have missed it, but what is the criterion to decide that a spiking neuron is one of the ones that triggers the seizure?
So I don't know if I necessarily laid out any criteria to say if it's triggering the seizure, but for ordering them as far as onset time or onset rank, I gave them a threshold. So, basically when did they cross threshold? And that was set on a cell by cell basis as a mean plus 3 standard deviation from a baseline period. We also considered that how we set threshold, is very critical for what their onset time is. So I went through a couple of variations, giving constant thresholds, kind of pre-processing the calcium data in different ways of filtering it. And so I've got a couple different iterations of how we define threshold, and it was always the same result, that we had this very stochastic variable recruitment seizure to seizure. If that answers the question.
Okay, cool. I'm going through the chat right now. It looks like we have 3 questions that are kind of all related to each other. So I'm going to try my best here to give them to you.
Okay.
So it's Scott Barabin, Jamie McGuire, and Ethan Hollingsworth. And so they're kind of asking the same thing. So Scott's question is, as the organotypic slice preparation is a reduced prep, only a few hundred microns thick and not an in vivo prep capturing the entire network, is it really possible to conclude that hub cells, which may not be in these cultures in the first place, don't play a role in ictogenesis? And then Jamie had a similar question, talking about the technologies emerging to image large number of cells in vivo, which may make it feasible to determine whether similar mechanisms are at play in vivo. And so she was wondering about the relationship to in vivo.
Mm-hmm. Yeah, no, those are great questions, and I fully acknowledge that these are organotypics. It is for sure a reduced preparation, and I would say I can't conclude that in vivo there may be hub cells, and they may have not ended up in our slices, or their processes may have been severed. So I can't say, and I wouldn't say that there's no role, but what it does suggest is that they're not necessary. So in this preparation, lots of seizures, lots of spontaneous seizures are being generated without hub cells. And to Jamie's point about shifting in vivo, that is kind of where we're going next. Organotypics give you, a lot of ability to really, image the whole network and get a lot of detail. And from here, we're really hoping to use that to guide moving in vivo, because I think it's true. Any conclusion that comes out of organotypics is, has to kind of be moved in vivo to really convince myself and to believe, how generalizable this is to other in vivo epilepsies.
So I have a similar type of question. So it kind of goes along the same theme. So I'll just jump in right here. In a lot of the trauma models, you lose interneurons. And in your slice prep, it looks like you have a lot of them there. There's no regional loss or anything like that. And what role do you think, that's having? Because we often equate interneuron loss with, increased risk for seizures.
Yeah. So first I'll say there may be interneuron loss, and there is sort of a lot of initial cell death from the slice preparation, but there's not, there's clearly a ton of interneurons that are still there, and there's not ongoing cell death. And they don't seem to be preferentially lost over principal cells. So again, I would say that it means that you don't need to have loss of interneuron, that there are ways that seizures and epilepsy can develop that's not dependent on just loss of interneurons. And again, I think this model may only be relevant to certain types of epilepsy, and it's not going to necessarily generalize to others, but it does, I think, show that you don't need to have a loss of interneurons to really drive seizures.
Okay, so I'm going to skip Chris and I'm going to come back to him. But first, John Hugenhard put in a point. He says, great talk. The issue of generality of results in different seizure models will be raised by reviewer number 3.
Yeah.
So enjoy. Okay, so then Chris asks, he says, Chris Dula, it's striking how much your recordings look like some subclasses of human seizures, low voltage, fast, sentinel spikes, etc. How much do your imaging traces mimic the EEG on a class by class basis?
Yeah, so that was something I was very interested in, and it's a little, because the majority of this imaging is just calcium imaging, and then we started doing some with the paired LFPs to really start to relate this calcium activity to the electrical activity and to the EEGs, because I think from a clinical point of view that would be very interesting to people, because a lot of the ways that seizures are categorized is based on the EEG. So I do kind of think of that, the low amplitude fast, as the correlate to low voltage fast, and there are seizures described in patients as either sentinel spike or lots of other different types of rhythms, and it's really being described on the basis of how the EEG looks. But yeah, I think that's something I would like to expand on, and for what we saw here was that there wasn't necessarily a subclass that had a different, the onset looked rather similar in the measures we were looking at, but it's something we've considered about maybe using these slices, testing if different pharmacologies work better in one type of onset or another, to really see if we can use this in a clinically relevant way. So yeah, I think they do mimic them. We've spent some time looking at that. And as far as, I guess what I was interested in is we saw this evolution in the types of onset, and I've been searching to see if that's something seen in patients. It's a little bit unclear because most patients don't necessarily have, I think, enough of the recordings, but there are patients who have more than one type of seizure onset. So that also makes us kind of think about that, because this is kind of getting away from your question, but I think there's a lot of speculation that if a patient has 2 different types of seizures, that it means they must have more than one seizure focus, whereas this might suggest that a single seizure focus, because the organotypic is, I think, small enough that you can really think of it as a single seizure focus, generates more than one type of onset. So I think in that way it could also help guide some clinical decisions.
Okay, so are there any more questions? Going once, twice? Okay, so there's a lot of questions. of course, if you're interested, you can get in touch with Lauren yourself. Jamie McGuire says, great job, Lauren. So congratulations. Great talk. And we do have it on the live feed. You can go back and watch it. If you're interested in that, you can see that on my lab's website. I will link to the YouTube live feed. We'll have it up at least all day. And then these talks I'm leaving up to the postdocs and trainees, whether or not they want to leave them up on the live feed, or if they'd like to take it down because it's, contains data that they're sensitive about. But with that, yeah, thanks, Lauren. You did a great job.
Thank you.
And then we will meet again in a couple of weeks for the next round of talks.
Thank you.
Bye, everybody.